In the world of genetic testing, we're facing this really strange paradox. We can read our DNA faster and cheaper than ever before, which is creating these mountains of data. But here's the thing. More data isn't always giving us more clarity. Sometimes it's just giving us more confusion. I mean, just imagine it. You get a genetic test to figure out your risk for a disease. You're hoping for a simple yes or no, right? But instead, the result you get back is maybe. And that's the reality for a ton of people, and it's become a massive challenge for modern medicine. This chart just lays out the scale of the problem perfectly. It breaks down all the little genetic changes, or variants, in a huge clinical database. You can see the small slices for things we know are harmless or harmful, but then there's that enormous gray section. That represents variants of uncertain significance, or VUS for short. This huge sea of unknowns is actually the single biggest category we have. So let's dive into this flood of uncertainty. We really need to unpack why our amazing ability to read DNA has just completely outpaced our ability to actually understand it. And get this, the problem isn't just sitting still, it's growing exponentially. This graph here shows the number of these VUSs we're finding over time. And it's not a straight line, it's a crazy curve rocketing upwards. We are being swamped by a rising tide of genetic uncertainty. And look, this isn't some abstract problem for scientists in a lab. It affects real people, millions of them. Somewhere between 20 and 40% of people who get a genetic test walk away with one of these VUS results. That's a life-altering maybe that can leave whole families and their doctors totally in the dark, with no clear path forward. But what if we could flip the script? What if we could actually test all these genetic variants before they ever showed up in a patient's results? Well, a breakthrough technology is doing exactly that, and it is finally starting to turn this tide of uncertainty around. The solution is a technology called MAVEs. That stands for Multiplexed Assays of Variant Effect. Now, the key words to focus on here are multiplexed and massively scaled. Because instead of slowly testing one variant at a time, MAVEs let scientists test thousands, or even tens of thousands, all at once in a single experiment. It's a complete game changer. I mean, the old way was totally reactive. A patient gets a weird VUS result, and then maybe a lab spends months testing that one specific variant. MAVs are proactive. It's like, instead of trying to find a burnt-out light bulb by checking every single one in a skyscraper one by one, you just flip a master switch to see which ones work and which ones don't. The leap in scale and efficiency is just huge. So how does this actually work? Well, this workflow breaks it down pretty simply. First, scientists create a massive library with thousands of different versions of a single gene. Then they pop these variants into model cells. Next comes a stress test, like they might see which cells can survive when hit with a certain drug. And the final step is just to count the survivors and see which variants pass the test. It's an incredible way to learn what thousands of these genetic changes actually do to the function of a gene, all at the same time. Okay, so the technology sounds amazing in a lab, right? But the real question is, does it work in the clinic? Let's take a look at how MAVEs are turning all this data into real-world answers that are already changing lives. This is a perfect example. This family tree shows a history of cancer. So they did genetic testing and found a variant in the BRCA1 gene. That's a gene we know is strongly linked to breast and ovarian cancer. But here's the problem. It was a VUS, a variant of uncertain significance. Nobody knew if it was dangerous or totally harmless, leaving this family completely in the dark, just stuck. And this is where the magic of MAVs happens. Researchers had already run a massive experiment on nearly 4,000 different versions of this exact gene, so they could just look up the family-specific variant in their data. On these charts, you can see each variant is plotted based on a functional score. It's basically a grade for how well the gene does its job. The ones that work fine are on one side, and the ones that are broken are on the other. And the answer? It was crystal clear. The family's variant landed squarely in the loss of function group. It was broken. With this incredibly powerful new piece of evidence, that VUS was reclassified to likely pathogenic. And that one change, from uncertain to likely harmful, gave this family the clarity they desperately needed to make informed decisions about their health. And this wasn't just a lucky break for one family.
This is happening on a massive scale, across a whole bunch of studies. When MAVEs data was added to the puzzle, an incredible 55% of these uncertain variants were successfully reclassified. More than half of all those maybes were turned into clear answers, either harmless or harmful. This diagram just shows the whole thing so well. On the left, you've got this tangled mess of uncertainty coming from many different genes. Then, as the MAVE data gets applied, you can see those tangled lines just untangle, flowing into clear answers. Blue for benign, red for pathogenic. We're literally watching a tangled mess of confusion get straightened out into clear, actionable answers at scale. So, solving this VUS problem, that's already a huge win. But honestly, it's just the tip of the iceberg for what MAVEs can do. So the next big question is, where is this technology going to take us? The potential is just enormous. MAVs can help us pinpoint which cancer mutations will respond to specific drugs, making therapy more personal and effective. They can speed up the development of new medicines by mapping out exactly how proteins work. They can even help us predict how a superbug or a cancer cell might evolve to resist our treatments. But maybe the coolest part, the part that's really going to accelerate everything, it's how MAVEs team up with artificial intelligence. I mean, think about it. MAVEs generate the exact kind of massive, high-quality datasets that AI models need to learn from. And in turn, AI can help us analyze the MAVE results and even design smarter experiments. It creates this powerful feedback loop where each technology just makes the other one better and faster. So think about where we started. Just a few minutes ago, we were talking about drowning in a sea of genetic maybes that were overwhelming doctors and patients. Now we have a technology that can systematically turn that uncertainty into answers. We are, piece by piece, building a functional map of the human genome. So the final question isn't just about clearing up confusion anymore. It's about what new cures, what new treatments, what incredible scientific breakthroughs become possible when we can finally, truly understand the language of our own DNA.